Hi, welcome to our third video, which will talk about the generation of genomic data. In particular, we're going to look at the next generation sequencing technologies that are used to generate the sequences that you're going to be using in your project. So as I mentioned before, several years ago, there was a fundamental change in the way that we sequence DNA after decades of sequencing with basically the same technology. In part, that involved the massive parallelization of sequencing process. So we went from sequencing a handful of molecules at the same time to literally hundreds of millions at a time. And this generated a tremendous increase in sequencing output and a dramatic reduction in cost. These technologies produce more data, and in fact better data, and in many, many fewer steps. So it's now possible to go from a specimen, a hunk of tissue, or a pile of organisms to the DNA sequence of those in only a matter of days. These changes have really fundamentally affected the way that we look in different kinds of areas of biology. So you've seen this slide before, and this is just to show you once again that this all occurred around 2006, 2007 when it first started. These technologies first came online, and you can see that they've had this really dramatic effect on the cost of sequencing. That change in DNA sequencing costs and the rapid nature of generating sequence data has produced a whole bunch of new technologies or techniques for exploring different aspects of biology that are now really done with this next generation sequencing approach. I think the things that you might be most familiar with or might have heard of are things like RNA-seq, where instead of using chips and hybridization signals from labeled RNA on genes to measure gene expression, it's now most common to actually simply sequence the RNA from different tissues or different organisms and look at that, the number of RNA sequences that are then compared to the gene sequences, and we can basically digitally count the amount of transcription that's taking place from any particular gene in the genome. Other very interesting techniques are things like bisulfite sequencing, which allow us to, at single base resolution, map all of the patterns of methylation in a genome, or the really fun stuff where we're looking at the specific interactions of proteins in DNA sequencing using technologies such as ChIP-seq. This is a really fun sort of guide that was produced by Illumina in 2014. It's hundreds of pages, and each page is one of these new -seq techniques for exploring different kinds of biology, and it's just really wonderful. So you should download it and have a look when you get a chance. So we're going to talk very specifically about these technologies right now. The one that we're going to spend most of our time on and in sort of gory detail is the sequencing by synthesis technology from Illumina. And this technology is probably the one that's really contributed the most data, certainly, over the last several years, and is the leader for high throughput sequencing analysis and clinical analysis in many contexts. So, and it's also the one that will be generating, most likely for some time to come, the data sets that you will be using as part of your project. And it's very important to understand how your data is generated. And so I think it's worthwhile to have a look at this video, and there are other resources as well that you can have a look at. Right, so we're gonna talk about this, and it's a bit technical. If you don't have a strong background in molecular biology, if you understand how the polymerase chain reaction works and how DNA is copied and synthesized, then you'll be fine. And so it's really at that level, although there's a lot of terminology that we'll cover, right? And then before we move on, I just want, if for no other reason than because they're very cool technologies, I'll introduce you to Smart Sequencing by PacBio and the futuristic nanopore sequencing that's out there by Oxford Nanopore Technologies. Okay, so Illumina sequencing is this technology is the one that we're going to talk about for the most of this time. There's all sorts of wonderful YouTube videos out there that sort of have more flashy graphic and movie-like presentations of the technology, and then there's a really funny joke video that a friend of mine from Keene, Lauren Lonan, shared with us, and so I wanted to give that to you as well. So here's the basic process. This is just the very core idea of wanting to sequence a genome. And so, of course, you start out with a DNA molecule. And in fact, you start out with many, many, many copies of the DNA molecules. So the chromosomes, for example, of a bacteria, you might have hundreds of thousands or millions or billions of them in a tube. And those are fragmented. The first thing that we do is we break the DNA up into manageable pieces. And then down the left-hand column here, because of what we want to do with those fragments, one of the things we need to do is to typically repair them. And so you have to spend some time biochemically making sure that the ends of those molecules are going to be competent for what you want to do next. And what you want to do next is to take, for these unknown DNA fragments, you have no idea what these are, you want to attach known DNA sequences to both ends of those fragments, because those are going to be very important for the next steps in sequencing that we see. So in this approach, we simply take these double-stranded DNA molecules that we bought from a place like IDT, or they come in the kits that are necessary for generating this, and they're called adapters. And those adapters are ligated to your unknown and chemically modified sequences that you have here from the previous column. Right? And so in the end, what you generate is unknown sequence in the middle, and then known sequences on either end of the molecule here. So this is a double-stranded DNA molecule. And very importantly, I'll go over a couple of these sequences now. On the very ends of the molecule, you have these known sequences P5 and P7. And those P5 and P7 sequences have to match the platform that you're going to be sequencing these on. And one way to think about these, and you'll see why in a minute, is that these are really the targets for PCR primers. Because we're going to make copies of this DNA molecule in the future. And so those are and we need to know those sequences because we are going to add PCR primers in a very interesting way later on to this process. The other sequences are sequences that are known that are also going to be priming sites, but they're going to be priming sites, for example, for doing a sequencing reaction. And sequencing in the Illumina way is very similar to other ways where it's by synthesis. So you're looking at the nucleotides as they're added. And we'll go over that in detail in a minute. And then there are these other sequences that are important, which are sequences that are known. And we have, for every individual library that we make, for example, when we're making DNA sequencing libraries from each of your bacteria that you guys submit, each bacterium will have a different index sequence in it. So it's like a barcode. And that allows us to throw all of your genomes together in one sequencing reaction and then sort them out later using bioinformatics into the individual original sample names that you gave us. There's an alternative approach, which is actually the one that we will most likely be using for sequencing your genomes. And that's instead of shearing the DNA, using physical forces or ultrasound into these fragments, we use this really cool approach where you capture the activity of a transposase. So this is an enzyme that would typically be produced by a transposon, and normally would be involved in taking a transposon DNA molecule, which would be a complete molecule here joined instead of these two fragments, and inserting that transposon sort of semi-randomly in the chromosome, for example, of a bacterium. And so what the biotech companies have done that have developed this approach is to capture the activity of that transposase, and instead of having transposon DNA in there, it had Illumina adapter DNA sequences in there. And so that gets added here, and at the same time, because it's not connected, you can see what will happen is when this gets added, it actually breaks the DNA molecule up. So you accomplish both things. You end up putting known sequences on the end of a DNA molecule and fragmenting the DNA molecule up into manageable sized pieces. And in the end, you end up with exactly the same thing that we ended up in the other slide. We use this technique for your work because, one, it's a lot cheaper and it doesn't require always a large amount of starting material. And so now let's talk about how the sequencing reactions start. And so if you remember, we have these sequences that have unknown sequences in the middle, but known sequences on the end. And the sequencing reactions themselves take place on solid supports. So, for example, this slide, this microscope slide, essentially that you see over here on the right, is essentially an exact replica of the one where your sequences will be generated. And what it is it is in fact a microscope slide because microscopy is used in this process. And this clear area here, you can see the bench through the microscope slide here. This clear area is actually a channel that's inside the glass, and if you look really closely, you can see there's pores here, and there's a pore down here under this person's thumb as well. And so there's pores on either end, and so those ports allow for the passage of material through here, different solutions for this to conduct the sequencing reactions. And so the first thing that we do is we're going to take the library that you generated, we're going to denature it so that it's in single strands, and then we pump those single-stranded DNA molecules into this chamber, into each of these channels here. And on the inside of the glass channels, there are these PCR primers. So short single-stranded oligonucleotides, just like for regular PCR, right? The only difference is, and there's two different ones. I don't know if you can see the color here as well, but there's these orange ones and then these semi-green ones here as well. And it's kind of like a lawn. And so if you imagine on the inside of this glass, there are many, many hundreds of millions of these sequences. And the other thing that makes them different than a normal PCR, or the thing that makes them different than a normal PCR sort of reaction primer, is that they're covalently linked. That means permanently attached on their 5' ends to the inside of the glass. So they're not coming off of there. So what happens, just like in a PCR reaction, when you pump your single-stranded DNA into here, is that it's allowed to cool. And those sequences then, because these sequences that are on these PCR primers match those P5 and P7 adapter sequences, it allows these complementary strands to join and provides a competent 3' end on this primer sequence that can be then used to make a copy of that molecule. All of this area in here is the unknown sequence. And then on the other end, there's another adapter sequence. And so that copy is made, and you have a newly synthesized strand. Interestingly now, because that was synthesized from this primer here, which is covalently attached to the glass, when we heat this up and flow buffer through this system, you're going to get rid of the original copy here. And all you'll have is this one, but it's attached to the glass. Now, you could, so you have a single molecule, and this has happened hundreds of millions of times on the inside of that glass slide. Different fragments have annealed and been copied all over here, all over the inside of the glass on both surfaces. Okay, and so you end up with this single-stranded molecule now that has a big unknown stretch of DNA in there, I should say maybe a few hundred nucleotides long, right? And it's this, now it's attached to the inside of the glass. Now, what we can't do, unfortunately, is sequence that single molecule simply because of the detection limits of the system. And so there is a process that needs to take place that is really fun to think about and very cool. And it's simply an amplification step. Because we're going to not sequence a single molecule, but we're going to make thousands and thousands of copies of that molecule that are identical to it. Okay, and this is done on the glass, and it's basically a PCR reaction, but it's interesting to see what happens in a PCR reaction when the primers are attached to glass. What happens is, and this is called bridge amplification, is that this original sequence that you have, on the other side, it has a different adapter that is complementary to the other primers that exists on the glass slide, sort of randomly placed here. And so that when that sequence bends over and can anneal, then the copy of the other strand can be made in the next cycle of the reaction. And when this bridge amplification goes through several cycles, you end up with clustered groups of molecules that all have the same sequence, because they all derived from a single molecule that sort of seeded the reaction there. And because they can't go very far, they produce these clusters around every one of the original molecules that sat down there. And so what you end up with are these clusters that are very much like bacteria on a plate. You know that bacterial colonies, when they form colonies on a plate, those colonies are all derived from a single bacteria and are all essentially genetically identical to the original bacteria. And so we don't call these colonies of sequence fragments here, we call them clusters. And so once you have those clusters, then each molecule within the cluster is ready to be sequenced. And so the sequencing by synthesis reactions are conducted as follows. So think about this. So the glass slide is down here, and this molecule, now a single-stranded molecule, we get rid of all but one of the strands for each double-stranded molecule. And those molecules are blocked here on the ends. So this is the 3' end. And then we add a sequencing primer, which is very common in sequencing reactions. And so this primer is complementary to part of the adapter that you added. And this nucleotide right here is the first nucleotide in the unknown region of the genome that you're going to sequence, that unknown part of that fragment. Okay, and so this is the 3' end of the primer here. And so those sequences then are going to be extended, but they're only going to be extended one nucleotide at a time. And so you flow into the flow cell all 4 nucleotides and a polymerase. But the nucleotides in this case are special nucleotides. Okay, and so and this is a peculiarity and an important thing to keep in mind with Illumina, that each one of these nucleotides, and we can look at it on this next slide, has a couple of properties. First of all, they have a unique floor. So we can tell by color the difference between a G, an A, a T, and a C. Okay. The other thing that they have that's very important is that they are blocked on the 3' end. And so if you remember from your knowledge of DNA synthesis, the new nucleotides are added to this 3' OH down here on the base. And so because this is blocked, you can only add a single nucleotide at a time to each one of the fragments. Okay, and so if we go back here, what's going to happen is we're going to incorporate whatever the complementary nucleotide is here in this first round of sequencing. And that one will be specific to every molecule in that cluster. And so each cluster then, after that first reaction, will have a particular color. And so that's where the microscopy comes in. And then, in order to carry on with this, two things have to happen. One is we have to remove the color from that, and so that floor has to be detached. And then the other thing is that that sequence has to be unblocked, and so that the process can continue. And so what happens is, as we're going through this process, we are adding a single nucleotide at a time. And so this is what I call Illumina Stargazing. And so this is just taking us through, this is a small section, and this is, all, of course, all falsely colored, a small section of the inside of one of those microscope slides. And what we're going to do here, each one of these dots is a cluster of genetically identical DNA sequences. And so inside that slide, there are hundreds of millions of these clusters that the machine is keeping track of, right? And so we're going to follow two of these through this process, through the cycles. And so after the first cycle, the instrument takes fluorescent micrographs all up and down the slides, okay? And so this generates a tremendous amount of information. And then that information is processed to determine that at this cluster, in this spot, in this particular case, this was a C. In this other one, this was also a C in this case. And you can see that there are other nucleotides that are being added. And then that color has to be washed off in that cycle. And then we go through a whole nother round of synthesis to add one more nucleotide. In that next cycle, in this top sequence at this particular position, we added an A. And then in the next cycle, we added a T and a C and a G and a T. Every one of those cycles, the microscope slide is flushed, the floor is removed from every one of those clusters, and the 3'OH is unblocked, and then you go through the process of adding another single nucleotide. And so this is the reason that sequencing with Illumina takes quite a while. Every one of those cycles takes multiple minutes to go through and image process, and so it's a laborious process, and luckily it's all done robotically and computationally. So that's the fundamental process for doing this Illumina sequencing. I think there's a couple of important things to keep in mind. There is the possibility, and in the way that your data will be generated, the number of cycles that you can go through determines the length of the sequences that you will have. And so because of the way Illumina sequencing works, every one of the sequences that you have is exactly the same length because it's determined by the number of cycles that exist. So in most cases for your data, we will allow the machine to conduct 250 cycles of sequencing. And so you will have, beginning from the first nucleotide of unknown sequence, 250 nucleotides of your presumed bacterial sequences. And then what we do is we allow the instrument to sequence from the other end of each of those fragments. And we will sequence another 250 base pairs from the other end of the sequence for you. And in this way, our sequences are what's called paired end, or PE sequences. And so your sequences, under most circumstances, every one of them, and there will be a lot of them, will be 250 base pair paired end sequences. So I just wanted to quickly introduce you to a couple of the other technologies. And so one of the technologies that's out there is often referred to just as PacBio, but it's also referred to because of its the way it works as smart sequencing for single molecule sequencing. And so this works in a very interesting way. If you look at this diagram, and there's some really interesting videos. This is actually single molecule sequencing, and sequences very long molecules that you provide to it, and sequences them in real time. So the SMART is single molecule real time sequencing, and the way that it works is it has at the bottom of a very large plate full of wells called a picotiter plate. It has polymerases tethered, and those polymerases are next to a detection system, and the polymerase incorporates, as DNA polymerases will do, nucleotides into a growing strand. So again, it's synthesizing DNA and keeping track of what nucleotides are being added to the growing DNA strand. Except unlike with the Illumina sequencing, this does this in real time. And so every time a nucleotide is added, and the nucleotide has a specific floor attached to it, it registers the color of that new nucleotide that's added. And it goes at the speed of DNA polymerases, which is very fast. So this is also a very fast technology. The other difference with this technology is that it is not limited in the same way by the sequencing lengths. And so it's possible, this is a long read sequencing, one of the long read sequencing technologies. And so it really has an advantage over Illumina when the length of the sequences are very important. And we'll talk about that probably later in some other videos when that's important. The downside to PacBio, frankly, is that it's not as inexpensive. And the sample preps, of course, to have long DNA and take advantage of that are more laborious. But when you need long sequences and you have the money, PacBio is a real, can make a big difference. The other sequencing technology that's out there that is not as far along commercially as PacBio is so-called nanopore sequencing, which is really almost magic. But similarly sequences individual molecules, and the way it does it is that it passes single-stranded DNA molecules down through a protein pore in a membrane and measures the change in the electrical properties of the membrane as the DNA molecules are passing through here. And it relies on the fact that the effect of those different sequences of DNA that pass through here and register on the pore, and the effect of the electrical properties of the membrane are unique for different sequences. And so this is a technology that has several potential advantages. One is instead of a machine that's the size of a small car, you have a machine that looks like this, which is actually that is a USB that plugs into your computer, and it can produce sequences and does produce sequences that are even longer than the PacBio sequences, and so very, very long stretches of sequence. Okay. The problem is with these, both PacBio, but PacBio has figured out ways to overcome this, is that these sequences often include long stretches where the sequences are not accurate or non-existent in the sequences. And so there's a lot of active work going on here, and so I'm sure that these will be wonderful additions to the technology in the future.